symmetrical repeating pattern. So they actually don't typically have that really highly negative charge. The proteoglycans do. And the proteoglycans are the ones, because they have a more negative charge, they're gonna pull water in. So this is the jelly portion to our joints. So this is where the proteoglycans and support of the proteoglycans will help some good joint maintenance, and especially in patients that have joint degeneration. So the glycoproteins, mainly we're gonna see on the cell membrane. We're gonna see a lot of the peptide hormones we'll get to in the next lecture, which is why this is kind of rolled into this section of material. And collagen. Collagen has a bunch of glycoproteins. So this is the, if you look at a glycoprotein sticking out in this little cartoon, he's, kind of folded. This is the protein piece that is green, and then we've got a couple little sugars in random sets branching out. Whereas the proteoglycans are a little bit more structured, and remember we had this term called the brush bottle appearance. So this would be a proteoglycan here. So here's just like a little rod in the middle, and you can see how these are very rigid, very structured, very sequential, kind of repeating units. And those are the sugars that pop out. So when we have a whole aggregate of proteoglycans, this is what composes our joints, right? Kind of the jelly portion and the waterous portion of our joints. Okay, so we said that is a short chain oligosaccharide, and we've got a bunch of amino acids that might be in there. So the proteins, the amino acids, are the ones that are holding on to that sugar modification. And this is where this is. We talked about the difference between putting sugars on reversibly and putting sugars on irreversibly. So the term that is natural and the most frequent way in which we modify proteins so that they look different, act different, give off different signals and stuff like that is glycosylation. And that's normal. That should happen all the time. It's reversible, and it does a job, a function. When we get to glycation, that is the forever put of a sugar, so it never comes off, it's irreversible, and then that's the problem. So why are we throwing sugars on proteins? Well, a lot of times the sugars will kind of signal that, look, I'm not done yet, so don't come and break me down. So it protects it against denaturing and proteolytic degradation, so it kind of stabilizes it. It also can help it get through cell membranes. It can help it get onto the cell membrane, so that's like the solubility portion. And if it is sticking on the outside of the cell membrane, that's usually because it's serving a purpose to signal something. So this chart is from your book, and we're not going to look at all of these, but it kind of shows you the different types of sugar that are put on to our different types of amino acids. So we've got a fructose sugar, not an R, so it's not fructose, it's known as fructose. And in nutrition 1, we'll talk about that a little bit, but I can actually pull up. There's this sugar known as 2FL, which is a fructooligosaccharide, and look at it. And it's a sugar that has, I don't know why a Chevy popped up, a Chevy Silverado. Oh, is that a 2FL? Oh, interesting. That's what sugar. Yeah. Let's see if the structure will pop up. And mucin. There we go. I think so. So this is a proteoglycan, or a glycoprotein that is, that looks very much like mucin in the gut. And where we find 2FL, that fructooligosaccharide, is very high concentrations in breast milk. So that's why breastfeeding has been shown to help to mature and protect the baby's GI tract until it can really mature to fully about 6 months or so. So there's an awesome little sugar that's naturally put in there by mom, and it protects. It doesn't really show too much of it, but what it does is that the 2F, so this is what mucin looks like, right? This is a glycan, so it's a protein sugar. And your epithelial cells, your goblet cells, will make the mucus for you. The mucin that protects the top of the GI tract. Well, when a baby drinks breast milk, or a formula. So formulas now in the past probably 5 years have been supplemented with 2FL, which is super interesting to me because that means the formula company is kind of keeping up with natural progression, and that'll actually protect the GI tract as well too. So either exogenously in formula or exogenously in breast milk, but it's also, like, Standard Process has it's called 2FL, and it's for kids, but it works really well for adults that have inflammatory gut problems as well, because it looks like mucus, so it does the same protective natures. So super interesting. So that's our fructose. Other sugars we got going on are galactose and glucose, and those are all the ones that we learned about that are in our, hyaluronic acid and chondroitin sulfate and things like that. So you'll notice that we got a couple of bonds going on here. You see how we referred to this very, very briefly last trimester, but there's this thing called an N and an O linked glycoprotein, and those are the types of bonds that the structure uses. And why do we care? Well, if you're N-linked, then you are a glucose with an N-acetyl, like an NAC, and it's typically attached to the amino acid asparagine. And these guys are either stuck in the membrane or they're rolling around in the plasma doing a job for us. If you have galactose, and you have serine or threonine, this is typically what's put either in like a mucus barrier or in your joints. So because if you guys remember, this is your amide, right? So when you have aspartic acid or aspartate, and you throw an extra ammonia group on there, it makes asparagine. And these guys have hydroxyl groups on them, serine and threonine. So if they have hydroxyl groups, that makes sense that they're gonna pull in more water than someone that has an extra ammonia, because it's more polar, it's more negatively charged. So the structure of that bond with that amino acids means that we have a lot more water being pulled in. So this is just a chemical look at it. So there's our N-link, there's asparagine, and then over here is our serine, and we've got glucosamine or we've got galactosamine, and we just have an acetyl group on there. That's why it's called N-acetyl. So the first thing we're going to talk about when it comes to the membrane-bound glycoproteins are these things known as GPI anchored, and that just means that we have a sugar. We've got this phosphatidylinositol, which is a phosphate and a fat. So we can eat inositol and we can take it as a supplement. And this guy known as an ethanolamine. So I do have a picture. Okay, I'll show you the picture in a second. But why do we have these glycoproteins? Like, why do we have these specific GPI guys? Well, they help with actually moving. Remember last time we were on Zoom, but we were talking about how like proteins will come together to create like a little like working section of the cell membrane, like in the lipid rafts, and then they'll move away. Well, the GPI anchored helps the proteins move around the membrane. They might actually do a lot of signaling, which is what we kind of know a lot more about. Like we'll talk about this a couple of times in like the insulin and glucagon lecture. And then they can actually help us to target proteins in the epithelial cells. So, and what that means is that it kind of orients the cell to say, okay, this is my apex and this is my basal layer. So cells kind of stay put. So this is what it looks like. This is an example of actually acetylcholinesterase linked to the plasma membrane of a red blood cell. So here's our phosphatidylinositol. So here's the membrane, here's our fats, here's a phosphate. And then the inositol is on top of the phosphate and connected into the fatty membrane of the cell membrane by another fat. Then we've got our glucosamine. Then we've got a bunch of sugars. Here's our ethanolamine. And then we've got our amino acid and our protein. So that this piece in particular is specific to acetylcholinesterase. So that's a receptor that works on your plasma membrane red blood cells, right? So your red blood cells, when they react to acetylcholinesterase and they do their job, this is how the cell signal gets in. So that's just an example. Okay, so Another reason why we might care as chiropractors, other than it just seems like a bunch of words, right, is that we actually will change in our bodies the different types of sugars we have. So, going back, looking at a serine, for example, remember the other one could be threonine, is an O-linked, an N-linked is asparagine. So when we have our procollagen is mainly the O or the N-linked guy. So he's a little more rigid, right? He's got asparagine, so he's got a little less water to him. He's a little more structured. And as we start to mature collagen and then put them into, say it's collagen especially that's going into like your vascular system, like your endothelial system, right? Things like that. Or it's going in and around organs to support it. They're going to remove more and more N-linked bonds, and they're going to put in exchange O-linked bonds as they mature. So if you have more fibrous tendon structure, so if you have a tendon, right, and you want that to be more fibrous and more kind of solid in its structure, you're going to leave a little more of the N-links in there. If you want something that's more in that mesh network, like your kidney, basement membrane, things like that, then you're going to take more of the N's and put more of the O's in. So, just kind of a neat thought, whether or not it kind of resonates with you right now or not, but it's neat to think that our body is not stuck in like one pathway, like this is how we make collagen, and this is the collagen that goes everywhere. No, it's not true. We're going to hydroxylate and make procollagen, and that's the base work for everything. And then, depending on where it's going and what the function is, we can actually change the chemistry of it by changing these bonds. So, who's really, really mucousy is mucin. So, who has a high amount of O-link glycosylation is him. So, he actually has another special guy, sialic acid, and he's really negatively charged. So this is going to do one of two things: keep a really big open space, so it doesn't really fold into like an actual like protein sense, and create a lot of water, so very viscous. Cancer cells can actually make mucin to mask their cell surface antigens. So they can start to mess with N versus O-linked bonds, and they're going to create more of an O-linked structure, which creates more water, and that mucin or mucousy layer actually masks the outside of the cell, so your immune system can't say, just looks like a big mucus blob, and white blood cells go right by without noticing that that does not look like, remember those MCHs or MHCs 1 and 2s, right? And if the cancer cell was a cancer cell, then they displayed a different I'm not myself flag on the outside, those major histocompatibility complexes. So this mucin masks that. Then what they can also do, which is crazy, is that they can change the glycoproteins like the MHCs on the outside altogether. So it can not only say I'm not myself, but change the ones that adhere to cells, and that leaves them a little more rolly, right? So they can move around, and this is how metastasis occurs. So when you go rogue with the appropriate formation of your glycoproteins, you obviously can create problems. So how are we making them? Well, we're going to start making them in the rough ER. We don't need to know any of these specifics, but we're going to, once we start to make the proteins, we're going to throw a bunch of sugars in the lumen of the ER as it goes towards all the ribosomes and gets modified. And then finally, we've got the little guys, right? So once it runs through the ER and it's getting folding and structure development, like baseline structure development, and some proteins or sugars are getting put on it, then it's going to pop off in a little vesicle and head towards the Golgi apparatus, and he's going to do further modification of a lot more of the O-links. So he'll start to change it a little bit more into that watery substance. So this is kind of bringing us to our first like nutrition thought. There's proteins that are hanging out in the rough endoplasmic reticulum, and they're known as chaperones. So they're literally there to help the new proteins along. They're going to help them fold, they're going to help the sugars go on and things like that. So in us, we have a couple of really specific guys called calnexin and calreticulum. And those names are not as important, but the class that they're a part of are known as lectins. So has anybody heard of lectins before? Yeah.
Yeah, I follow a channel called Sarah Kleiner Wellness, and every single video is about leptins.
Yeah, and what was his name? Well, it's going to be on here. Who's the plant paradox guy? Steve Gundry. I don't know if she speaks about him a lot too. So yeah, he talks a lot about lectins. So we're going to watch a video because way back in, oh boy, I was in school learning. So we're talking about like the 2000s. And I'm so old, yeah. I graduated in 2007, so how old was everybody in 2007?
7.
Oh, you guys are so cute. You was 4? I was 3. You was 3? I feel like baby size.
I feel old.
Yeah.
Yeah, I was 7.
That was graduate school, so I graduated high school in 2000. Yeah.
I told you yesterday, you said don't graduate high school the year I was born.
Yeah, that's when I graduated.
That's
Is that
what
yours?
you
Yeah,
said.
you're stone. Isn't that weird to think that like I couldn't drive in and your head's going?
Sorry, we were on 2 different conversations. I'm old. Yeah, so I'm feeling well, so you're stone. I'm like, wow, that's horrible.
So lectins are like a hot topic since then. I had a professor who was teaching me that she was really especially about nutrition. But like she was all about you can't eat certain plant-based foods because of lectins and they destroy your gut. So the author that wrote, it's called The Plant Paradox, Steve Gundry, and it's very controversial. So we're gonna watch a video about whether or not he's like removed all lectins. And I think we're gonna talk, some people do really good with removing lectins, but other people, it doesn't really matter. So what are they? The reason why they're a problem is they are proteins that, because they are those chaperones, they're going to look for certain glycoproteins. So what they're supposed to do is purify, refine, recognize, all that kind of stuff and be a helper. But when you find them in a natural source, like you have lectins inside you that help you in your rough endoplasmic reticulum, but so do other plant sources like plants and foods we eat, and animals will have them too. And if you are a plant lectin, you could possibly create this problem with, they used to be called phytohematogglutinins. And what they do is they recognize our red blood cells, and instead of helping, they're reacting with the cell surface of our red blood cells, and they're creating them to clump, which is the glutination portion of it. And that's why, it's a natural response to some plants so that you don't eat them, right? Or a bug doesn't eat them, or a cow doesn't eat them, or something like that. It's just their natural defense mechanism. But the idea behind it is that, if it's their defense mechanism, we shouldn't really be eating it, because then it'll damage our cells and our GI tract. So we can strip the intestinal mucosa, we can agglutinate our epithelial and red blood cells in there, all kinds of problems. So that's the thought behind it. So we're gonna watch, forget if we watched one of her videos before. This is Dr. Laura, and she's really good. She's actually, I like her, she's got a whole little channel, Nourishable. Have you watched that before? Any of those? Yeah, Nourishable. But she is a biochemist and a nutritionist. Oh, I like her, right? So we're gonna watch this about her kind of debunking a little bit, and I like that sometimes she's a little harsh on the people that she debunks, but she's just showing the science to it. So we'll probably over the course of between this and nutrition, watch 3 or 4 of her stuff. She also, now I'm going on and on about her, a little bit of a hero. But if you go to her Nourishable page, if you're ever interested, she has a bunch of videos that are specific to science, nutrition in the microbiome. She actually has, it's probably the lectures, so digestion, metabolism, cells. This is these are really good board review videos as well too. So, said what? Emailed her, probably a fecal analysis, yeah. It's just got some good stuff.
In 2017, cardiologist Dr. Stephen Gundry published the book The Plant Paradox, The Hidden Dangers in Healthy Foods. In his New York Times bestseller, Gundry proposes that a lectin-free diet is the cure for nearly all health woes, including obesity, type 2 diabetes, heart disease, and autoimmune conditions. This lectin-free diet eliminates all legumes, whole grains, most fruit, and vegetables that are botanically fruit, like tomatoes, eggplant, and zucchini. Dr. Gundry claims to have cured over 1,200 patients with his lectin-free diet. But what is a lectin anyway? Does research support that lectin-containing foods are actually making us sick? Let's science it. Welcome to Nourishable. I'm Dr. Lara. A lectin is a type of protein that binds to carbohydrates, like Lego pieces. Lectins bind to carbohydrates that decorate the outside of cells to help cells communicate with each other. Lectins are ubiquitous in nature. They're found across animal, plant, and microbial species, and yes, even inside of you. Some plant lectins have evolved to help protect against predators, like insects and animals. When these plant lectins bind to cells in the gastrointestinal tract, they make the predator feel sick, so it will avoid that plant in the future. Lectins are found in many plants, including the legume family of beans, peanuts, and soy, the skin and seeds of fruits, and fruits parading with vegetables like cucumbers and squash. The lectin wheat germ agglutinin is especially high in whole grain products. In order to understand why Dr. Gundry proposes that lectins are bad, we need to have a little chat about the intestine. Inside our guts, we all have a large population of bacteria that make up the microbiome. Our microbiome is nourished by what we eat, and in turn, our microbiome can help keep us healthy. Dr. Gundry proposes that lectins cause unhealthy bacteria to take over the microbiome. This unhealthy microbiome causes inflammation in the gut, making the intestine very leaky. A healthy intestine is like a solid wall of security guards that is very specific about what it allows into the body. But a leaky gut has holes that allows anybody to enter. Gundry proposes that lectins bind to intestinal cells and then enter the body through a leaky gut. Once inside, he says these lectins mess up our hormones to drive obesity, type 2 diabetes, and cardiovascular disease. He also states that these rogue lectins tell the immune system to attack its own body tissues, causing autoimmune diseases. Those are some pretty extraordinary claims. So what is Dr. Gundry's science? There have been no human studies investigating this lectin-free diet. Dr. Gundry's claims of reversing disease in over 1,200 patients haven't been published in the medical literature. The book cites many cell culture and animal studies and then directly applies these findings to humans. But humans are way more complicated than cells in a dish. For example, Dr. Gundry claims that wheat germ agglutinin from whole grains causes obesity and insulin resistance, factors which drive type 2 diabetes. Now, there was a study back in the 1970s that put some fat cells in a dish, bathed them in a bunch of wheat germ agglutinin, and showed that it binds to the insulin receptor and prevented fat breakdown. But this was way more wheat germ agglutinin than could ever be eaten, and these findings haven't been substantiated in humans. In fact, quite the contrary, because people who consume more whole grain have a lower risk of type 2 diabetes. Since I couldn't trust the research in the book, I decided to do my own nourishable analysis of lectin-containing foods. There are some rare examples of highly problematic lectins. If you eat raw kidney beans, you'll experience food poisoning from a lectin called phytohemoagglutinin. But this lectin is denatured with cooking, so it isn't a reason to avoid all lectins. For the general population, lectin-containing foods are very healthy. Long-term human studies have shown that people who eat more fruits, vegetables, whole grains, and legumes live longer and have a reduced risk of chronic disease. Lectin-containing foods support a healthy microbiome. Legumes play a foundational role in all the diets of the healthiest super-ager populations around the world, like the Okinawans of Japan, the Sardinians of Italy, and the Seventh-day Adventists in California. So does that mean that lectins are healthy? For most people, lectins themselves are pretty innocuous. Not bad, but not specifically beneficial by themselves. But keep in mind that we don't eat nutrients in isolation, but rather we eat whole food. Just like when you're evaluating a potential romantic partner by their friends, you can evaluate a nutrient by the company that it keeps. Lectins themselves are pretty neutral, but the other nutrients that accompany lectins in fruits, vegetables, whole grains, and legumes are packed with benefits. All of those vitamins, minerals, antioxidants, and fiber work together synergistically to support health. There are specific groups who are sensitive to some types of lectin-containing foods. For example, people with celiac disease and non-celiac gluten sensitivity need to avoid gluten from wheat, but other lectin-containing foods are fine. If you're experiencing gastrointestinal distress and you suspect a food sensitivity, consult a gastroenterologist and registered dietitian nutritionist. They'll run diagnostic tests and a structured elimination diet to help figure out your specific triggers. My nourishable conclusion is that Dr. Gundry's lectin-free diet makes some pretty extraordinary claims. Extraordinary claims require extraordinary evidence, and that evidence just isn't there. In my opinion, the Plant Paradox book is a compelling piece of pseudoscience. I consider it irresponsible to promote a lectin-free diet as a cure-all without the research to substantiate the claims. The consensus of research to date supports that lectin-containing foods like fruits, vegetables, whole grains, and legumes are the foundation of a healthy diet. And now it's time for a lectin-full lunch. That's what science tastes like. Thanks for tuning in to...
She cooked him.
She did cook them, yeah. Pseudoscience. This is what a lectin high and low diet would look like. So like she said, a lot of the high lectin foods are those things that carry a lot of fiber, right? And we've got, grains and oats and rice and legumes and nuts and seeds, and a lot of them are very high lectin. So I would agree. I really like her analysis, and I remember reading the Plant Paradox, and I was like, makes sense, I guess, but I don't necessarily know if it's a good idea to pull all kinds of grains and fiber sources away from everybody. But that being said, I wouldn't throw Dr. Gundry out with the bathwater kind of thing. He wrote a book recently in the past probably 4 or 5 years called The Energy Paradox, and that one I thought was really good. That was a lot less extraordinary, where it's like everybody has to do this, and more like this is how you support mitochondrial health. So that's nutrition too. We'll look at that, his videos, and that science when we get to nutrition too. So something to think about, there's definitely people, what this mimics, which we've talked about a little bit, is it mimics like a low FODMAP diet. And so you're pulling a lot of the fibers out that potentially bother people that just have an inability or a harder time digesting a lot of high fibrous foods, which is the lower FODMAP diet. And that has a lot of good evidence behind it with like IBS and inflammatory bowel diseases, and just helping with symptoms and regression of the inflammation, mainly because you're pulling stuff out that's hard to digest. But does that mean that everybody does it forever? I'm not sure, right? So there's some people that will naturally kind of go to a lower lectin food just because as soon as they eat beans, they get so bloated. And that doesn't have to be forever, then you help them with digestion. So get them to, because beans are incredibly important for health.
So what foods are left? That seems like it just funnels you into carnivore.
Yeah, I mean, there's no, like, you can't do the legumes or anything like that. You can do rices, you can do, like, greens and stuff like that. Vegetables, yeah. So, not carnivore, more like paleo almost. Yeah, Lexi.
Is Whole30 the same as low FOD, or is that?
Yeah, yes it is, yep. So it's like a Whole30, yep. Yeah, I would say probably there's not much different between this low lectin and a low FODMAP diet. So, because what it's taking out, the FOD is fructooligosaccharides and disaccharides. So you're taking out a lot of the really heavily starchy stuff, like your beans and lentils and peas. Yeah, I mean, overall, for whatever reason, I do better on a lower lectin, but, forever, I've always had digestive problems. They're so much better than they used to. But a lot of people that just kind of don't digest well do better on a low lectin diet. Okay, it's a little early, but let's take a break, because this is like a whole nother section that we'll start talking about. So let's come back in about 15 minutes. Well, let's do 8:45. Come back at 8:45, about 13 minutes, and then we'll finish the session.